We want to hear Elixirgen scientifics, Dr. Tetsuya Tanaka. Elixirgen is Baltimore-based company, and the campus in Johns Hopkins University. And Dr. Tanaka currently serves Director of Molecular Research in Elixirgen. He has a Ph.D. in Development Biology from Osaka City University and postdoctoral research in NIH, Institute of Aging, and research in University of Toronto. And he joined before he joined Elixirgen, he was a professor at the Notre Dame, research associate professor. And he was extremely experienced scientist and published numerous books. He sent me his CV, and it was more than 20 pages of all these publications. So I don't think I'm going to cite all of them today, but he's very seasoned scientist with specialty include developmental biology, embryonic stem cells, and genetic engineering and molecular cloning. Dr. Tanaka, please tell us what you have to say with Elixirgen today.
Thank you, Chio-san, for a kind introduction. And then we'd like to thank the organizer of this meeting for giving us this opportunity. We are very excited to share our technology with you today. And then we are trying to accelerate everyone's research progress using our technology. So our company is located in 2 sites in worldwide. U.S. headquarters are located in Baltimore, Maryland, and then we do most of the research and development and manufacturing activity there. And then we recently opened a Japan branch at Kawasaki City in Japan. And here we will do mostly sales at this moment, but eventually we will start the R&D activity also. Now, at the core of our technologies, iPSC technology exists. Now, the drop of blood actually used to tell a lot of patient conditions, but nowadays we can actually derive stem cells using a blood sample. And then this could be from patient or a healthy individual. And then those stem cells can actually serve as an unlimited supply of a specialized cell, like neurons, muscle cells, or liver cells. But there is a technical gap exists deriving those specialized cells using iPSCs, such as the currently existing protocols actually uses the long-term culture based, so that during the culture gets contaminated and one actually has to start over the culture again. Or that some of the protocols are very adapted to only particular type of iPSC lines and not applicable or useful for the other iPSC lines. So there is actually a technical demand exists in establishing a quick, easy, and reproducible method. And that's what we are trying to support. Then eventually make the everyone's research more accessible and accelerated. Then here I'm just showing a regular development of a mouse embryo, and then in 3 days, fast cell differentiation event occur. But the currently existing differentiation method is trying to recapitulate this difference development process, so that it takes a very long time to produce specialized cells. On the other hand, we have knowledge that within a cell, gene is activated, and then gene A activate, say, downstream target gene B and C, and then those B and C also regulates the further downstream genes, many set of genes. Then eventually it turns on cell type specific genes. So using this knowledge, we try to accelerate the differentiation by simply applying the introducing genes that specifically expressed in the specialized cells. And then I'm trying to summarize the our basis of our technology using this slide. Our founder, Dr. Minoru Ko, now has a laboratory at the Keio University, and in his lab did this experiment. In this experiment, 460 genes that are known to associated with the, some specialization of cell type is introduced into pluripotent stem cells one by one, and then the global gene expression profiles were examined, and then see which gene upregulation eventually leads to the similarity to the non-adult tissue or cell types. And for example, on the left top corner, there are a group of genes that showing a highly correlated expression of the brain tissue. And then if you look at the bottom of the, this matrix, there are a group of genes. Those upregulation actually shows a high correlation with the blood cells. Then using this knowledge, we actually take the group of candidate transcription factor and then make a cocktail. And then in the form of RNA. And then those cocktail is introduced inside a cell and by the transfection or by the Sendai viruses. Then by forced overexpression of those cocktail of transcription factors, we see if that that results in differentiation of our gene cell type of interest. In this example, like neurons or skeletal muscles. Then we repeat this process and then try to establish a protocol which is optimized to obtain differentiated cells within 1 week or at most 10 days. Now, in this slide, I'm showing a list of products currently available through our company, which is reagent to differentiate customers' iPSCs into neurons, skeletal muscles, or vascular endothelial cells, or using our iPSCs, we are also selling differentiated neurons to the customers. So now, in this slide, I'm showing some basic how our differentiation protocol look like. In this example, we are using Sendai virus that forcibly express one of those, some of those gene cocktail inside a cell, and then as you can see in the bottom phase contrast microscopic images, within 6 days, starting from day 4 to day 10, cells actually showing a dramatic morphological change. And then by day 10, the cells look like a mature neuron. And in fact, when those neurons are stained with the, some immunohistochemical method, those neurons localizes some marker genes. Then in this example, we are explaining how robust our method is. Here, we took the 3 different iPSC lines and then differentiated using exactly the same method. And then regardless of the iPSC lines used, our method actually successfully differentiate iPSCs into neurons. And then in the bottom, those neurons are stained by the immunological method and then showing nicely the marker of neurons. Then here, we are demonstrating how functional our neurons are. I'm not going to go into the technical detail of this method we use to validate functionality of our neurons, but this is called a patch clamp, one of the electrophysiological method to validate the electric activity of neurons. But this slide indicates that our neurons are actually functional electrophysiologically. So again, I'm summarizing our products and services available through the Elixirgen Scientific. On the left side, our products are listed, and the first one is a differentiation reagent kits. Using those our cocktail, customers' iPSCs can be differentiated efficiently within 10 days. And then so far, neurons and then skeletal muscles in endothelial cell kits are also available. And then we also sell cells differentiated using our iPSC lines, using those differentiation kits or protocols. And then we have an access to the California Institute for Regenerative Medicine iPSC bank, such that we can provide cells derived from, not necessarily from the healthy donor, but also the Alzheimer's disease, epilepsy patient, autism patient, you name it. And then currently we are almost ready to sell this assay-ready plates. We plate our neurons on plate for the multi-electrode array assays, and then we distribute this preceded MEA plate to the customers. So customers do not need to wait a long time to assay their chemical compounds for this assay. And then on the right hand, we also listing services available through our company. First one is a differentiation service. Using customers' own iPSCs, we receive those iPSCs and then differentiate into neurons, muscles, or endothelial cells on our side, and then ship back those cells in a frozen manner. And then we have a very well-equipped facility for particularly the neurological assays, or the microscopy is also a very well-equipped microscopy too. So then we can conduct those assays here and then provide the data to the customers. And then finally, if the customer's cell type of interest is not listed in our website or the list, we are happy to work with the customers to establish a unique differentiation protocol to differentiate those customers of interest cell type using the iPSCs. Then in the last 3 slides, I would like to show few examples that our technologies have been applied to model some disease. First one is Alzheimer's disease model. We use our method to differentiate neurons using healthy individual-derived iPSCs, as well as the Alzheimer patient-derived iPSCs. Then morphologically, those neurons look very similar, but at the gene expression level, we were able to identify some genes that are associated with the progression of Alzheimer's disease are highly upregulated. And in the second example, we use the iPSCs derived from epilepsy patient, and then compare the differentiation with the neurons derived from a healthy individual. And then morphologically, those neurons shows a distinct shape. And not only the shape, but also the electrophysiological activity. When the electrophysiological activities are measured using a multi-electrode array, we found that the neurons derived from epilepsy patient iPSCs show more activity. And this study was actually done in collaboration with RICO, which is also located in Kawasaki City. And then in the last example, we received iPSCs derived from a myotonic dystrophy patient, which was actually sent from one of our customers. And then we differentiate those iPSCs into skeletal muscle. And then if the muscles are derived from patient-derived iPSCs, the nuclei of the muscle show disease-associated phenotype. Red RNA foci was observed. But once the genetic mutation associated with the disease phenotype is corrected by the gene editing, those muscle cells derived from corrected iPSCs no longer show those RNA foci in a skeletal muscle nuclei. So this was actually nicely done. And then customer was very happy. So then he put the muscle into V shape, standing for victory. And he was able to publish this study in scientific journal. So to summarize, we offer this unique technology which derive the specialized cells using iPSCs in a quick and robust manner. And then those cells are functional. And then we are hoping that we would help customers to accelerate their research progress. Thank you for your attention. And then please leave any questions you have. I'm happy to answer those.